Decoding Our Genes, an illustrated lecture by Nobel Prize winner Venki Ramakrishnan. Dr. Venki Ramakrishnan grew up in India before moving to the US in 1971 at the age of 19. Following a PhD in physics, he studied biology for 2 years before he began his studies on the ribosome, the large molecular complex in all cells that translates genetic information into proteins. He moved in 1999 to Cambridge, England. For his role in the atomic structure of the ribosome and its complex with antibiotics, he shared the 2009 Nobel Prize for Chemistry. He is also the current president of the Royal Society and the author of Gene Machine, a candid memoir that describes both the quest for the ribosome structure and the human side of science. A warm welcome to Dr. Venki Ramakrishnan, Decoding Our Genes, an illustrated lecture.
Thank you very much. It's a great pleasure to be here. And as some of you may know, I've been a working scientist all my life. And this is really the first book I've written. And whether it falls in the fiction or non-fiction category is something that my competitors may have to decide after reading the book. So the question is, when people write scientific memoirs or biographies or histories, it's often written through the prism of hindsight. And really, that is not how things unfold, and that is often not the way things actually happened. There's a lot of revisionism. And in hindsight, everything seems logical, everything seems great, scientists are all rational beings, and so on. And that isn't true. And there was really one book that first changed the way science books are written, which was The Double Helix by Jim Watson. And this book is sort of in the double helix genre. And Richard Dawkins, when he described my book, fortunately said that I was the nice Jim Watson, which, given Jim Watson's news recently, I'm glad he added the word nice. So what are the 4 themes in the book? Well, the first is the main character in the book is not the scientist at all, but actually the ribosome, which is this amazingly complex and ancient machine. It's actually older than DNA. It existed before there ever was a gene to read. And it's a machine that reads the information in our genes to make all of us what we are, every life form. The other is what it felt like as an outsider. I didn't even get into any of the IITs, I should say. So I went to my local state school, and I went to a second-tier university in the US. So I was very much an outsider, an immigrant, someone who was not on the fast track of science. And what it felt like to suddenly be in the middle of a high-stakes race for this very fundamental problem in molecular biology. A third theme is about the human messiness of science. Science as an enterprise is beautiful. It's logical, it's empirical, it's self-correcting, it's all of those things. But scientists are not that. Scientists are humans, and so we have personalities and ambitions, we have egos, we have rivalry, competition, we also collaborate. And then we're also affected by external things like prizes and the desire for recognition. And then finally, there are some lessons from my own life in science, which perhaps aspiring young scientists might benefit from. It's too late for me to benefit from them. So I'll start off with the question of genes. Everybody thinks they know what DNA is, and because DNA is in the news all the time, we all talk about our genes. Oh, so-and-so has terrible genes. They have the gene, that gene from this parent or that parent. But if you ask them, what is DNA and what is a gene, and probe them just a little bit, you find out quickly they have no idea, okay? And what genes are, now this is a picture showing how many genes different organisms have. And it was thought that we had 34,000 genes, but actually we have closer to about 20 to 25,000 genes, which is not very different from the number in a worm or in a weed, Arabidopsis. And that should be, teach us some humility when we start thinking we're sort of at the epitome of life. But actually what a gene is information on how to make proteins. And if you ask what genes are, they're units, they're long molecules that contain information. And the molecule happens to be DNA. And this, of course, is the famous double helical structure of DNA. It has 2 strands, and each strand is a long polymer consisting of 4 different types of building blocks called bases. And somehow the order of these building blocks is important. You can think of these as letters in an alphabet, and so it's the sentence that is constructed using those letters that's important. Now, genes are used to make proteins, but proteins are also long polymers, but they're polymers of a completely different type. There are 20 types of building blocks in a protein, and they're called amino acids. Now, if you ask the average person in the street about proteins, they think of it as something in their diet that they need, to be healthy. You need to eat enough protein. Or they think of proteins as muscle. Muscles are only one kind of protein. In fact, proteins carry out most of the thousands of functions of life. So here, for example, is shown on the top is shown what a protein would look like if it were stretched out and drawn as a chemical structure of 20 types of amino acids. But you can also represent each amino acid by a letter. So next you can see a chain which is like beads, which each bead has a letter associated with it. That's just a schematic representation showing this protein has this particular order of amino acids. Now here is the miracle of life, and that is the order of the letters in a protein, the order of amino acids in a protein, itself contains the information to allow the protein to fold up into a particular shape. Now, this is almost like if you had a long strip of paper, and depending on what you wrote on that piece of paper, the paper would fold up, the strip would fold up into different shapes. That's exactly what happens with a protein, and it's really, in a way, quite amazing. Just to show you a couple of examples of proteins, the next shows a 3-stranded helix. Now, traditional Indian women will braid their hair with a 3-stranded braid. That's exactly what collagen looks like. Collagen is the protein that makes up our skin. And since I'm here in Chennai, you should realize that that 3-stranded helix was actually first proposed by G.N. Ramachandran, who was working at the University of Madras and worked with the Central Leather Research Institute, which is in Adyar. And then below that are a couple of proteins. On the bottom left is hemoglobin, which carries oxygen from our lungs into our blood to our muscles where it's needed. And on the bottom right is rhodopsin, which is a protein that senses light. And without that, you wouldn't be able to see, because that's what senses light and sends a signal to the cell that eventually goes to our brain. So it's just to show you, proteins carry out thousands of functions. They're in many different shapes, but they're all made using information in our genes. And that information somehow resides in the order of the building blocks in DNA. So the order of the 4 building blocks that are used in DNA somehow specifies what order the amino acids have to be assembled to make a protein. Now, here's the problem. DNA and the RNA, which is simply a copy of DNA which is read by the ribosome to make proteins, are both nucleic acids. So they have this 4-letter alphabet of 4 types of bases. But you have to somehow go from there to proteins which contain 20 different types of amino acids. So how do you go from a 4-letter alphabet of one type of polymer to make a protein which is made of 20-letter alphabet of a completely different type of polymer. That was the big puzzle in molecular biology when the structure of DNA was discovered. Nobody knew how DNA contained that information, how that information was decoded. And it took many years to sort that out. And basically, DNA is copied into a molecule called RNA. So you can think of DNA as a storage form and RNA as a working copy of any individual book. So DNA you can think of as a library, and you can think of RNA as the working copy of a particular book that's used to make a protein. And now you've got to take that information and make a protein. Now, if you simply took one base and represented that with an amino acid, you'd only be able to code for 4 amino acids. So clearly that wouldn't be enough. But if you read DNA 2 bases at a time, you could code for 4 times 4 combinations, and that would be 16 combinations. Still not enough. And what actually happens is that the information in our genes is read in groups of 3 at a time. And that unit of 3 is called a codon because it's a unit of the code. But how is it read? Well, Francis Crick proposed that there would be an adapter molecule, and he turned out to be right. That adapter molecule itself has another kind of RNA called transfer RNA because it transfers the amino acid to the growing protein chain. And it has at one end a group of 3 bases, and that recognizes the 3 bases on the codon. And if the recognition fits, then that transfer RNA's amino acid is accepted. And so then you would have a different tRNA or transfer RNA for a different amino acid, which would read a different codon and so on. So this shows you how you can get from the language of genes to the language of proteins. But it doesn't show you how the process would actually work, because it would be very unrealistic to think that this all happens by magic, by itself. Well, the way it happens was first shown when it was realized that proteins are assembled in particles inside the cell. And these particles were first seen as blobs in an organelle in the cell. And when the particles were isolated, it turned out that they all were about 300 angstroms, which is about 30 nanometers in diameter. And they all consisted of 2 pieces. If you look closely at the particles, you'll see that they all consist of 2 different pieces, a large part and a small part called subunits, the large and small subunit. And at some point, they were given the name ribosome. Now, when ribosomes were discovered, they were enormously complex. It turned out that each ribosome consists of 80 different proteins itself. Even though this is a molecule that makes proteins, it itself is made of proteins. But the largest part of the ribosome is made of RNA. So although it reads RNA, it itself consists of RNA. And that's a puzzle that I'll try to address later. And then it's made up of these 2 subunits that come together to make the entire organelle. Now, how does it work? Well, what the small subunit does is it binds the genetic message, which is shown as that long blue line in the figure. And the ribosome has 3 slots for it, which are called E, P, and A. The P is obscured because what's in the P site is the first transfer RNA recognizing the first codon, bringing in the first amino acid. And then what happens is another tRNA comes in, and if it happens to be the correct tRNA, it's accepted by the ribosome. What do I mean by correct? It means that the 3 bases on the tRNA have to match the 3 bases on the gene, on the mRNA, which contains the gene. If it doesn't match, the tRNA is rejected. But if it matches, the ribosome accepts it and then joins the first and second amino acid. And then the ribosome has to move. You see, it happens in 2 steps. First, the large subunit moves, and then the small subunit moves. And then the old tRNA is kicked out. And then the third tRNA comes in. And then if it's accepted, the ribosome joins the second and third amino acid. And then this process continues until the end of the gene is reached. So what you can see is the ribosome is reading the genetic information on the bottom, and on the top, it's stitching together the protein in accordance with those instructions. So that was sort of the state of affairs when I entered the field. So now I'd like to switch gears and talk about my own voyage and how I ended up working on the ribosome. So I was born in Chidambaram, as you know, but I left Tamil Nadu at the age of 3 and grew up in Baroda in Gujarat. And then I left at the age of 19 to do a PhD in physics in Ohio University, which is a small university in southeastern Ohio, because it was the only university that would actually give me a fellowship. And as you know, certainly in my day, a middle-class Indian couldn't possibly think of going to the US and actually pay for their education. So I went off to do my PhD in physics. That's the building where I worked. And I would say my years in Ohio were most notable because I met and married my wife there, rather than any great scientific discovery that I made. Now, what was the problem? Well, I started studying physics seriously, but as soon as I started doing physics research, I realized that I wasn't really cut out to be a physicist. And here are some of the problems. The problems in physics I found them to be very hard. And what I mean by that is not in a trivial sense, like exam questions being hard. Physics is a very mature subject, and it's quite hard to make really fundamental advances in physics. And in fact, when I was a graduate student, in the previous few decades, there had been really very few major breakthroughs in terms of changing our view of life. That has changed, with the discovery of dark matter, for instance, or dark energy and many other areas. But really, they're very few and far between. And I thought if I stayed on as a physicist, I would spend the rest of my life doing boring calculations that wouldn't really improve understanding. It would be sort of a tradesman-like way of doing science. And I didn't want to do that. So on the other hand, I used to read Scientific American, and I realized almost every issue had a major breakthrough in biology. And moreover, they seemed to be done by mere mortals. It didn't seem as though you had to be a real genius to do that sort of thing. And I also knew that many physicists had made this transition before. I mean, Francis Crick is one of the most famous, but also people like Max Delbruck or Max Perutz, who founded the lab where I work. And I thought of doing a postdoc in physics. I actually was offered a postdoc at Yale, but I turned it down. Actually, I was offered 2 postdocs, one by Don Engelman, whom I wrote to later, and another by Tom Stites, with whom I ironically shared the Nobel Prize. But I turned them both down and decided instead to go to graduate school because I needed a basic background in biology. So I went back to graduate school again. I should say at the time, my father, when I eventually told my father about it about a year later, he was not entirely happy that his son had gone back to graduate school after getting a PhD in physics. So anyway, I went to graduate school and I even took undergraduate courses. And the lesson here is, if you don't know something, you have to start at the beginning. You can't just sort of pretend you know something, and go on that way. And I'm very glad I did this because it gave me a broad foundation in biology, and I think it made me a better scientist than if I had gone and done a postdoc straight away. So I started learning some biology. I took undergraduate courses, tried to catch up on my background as much as I could. I did some lab projects. And then after a couple of years, I realized I didn't actually need a second piece of paper saying that you have a PhD. I felt the object was to learn biology, and I had learned that, and I was now ready to do research. And so I came across another Scientific American article, and it turned out one of the authors was somebody who had offered me a postdoc. And I said, Don Engelman, and I said, well, if you offered me a postdoc when I actually didn't know anything, maybe you'll be even keener now. And he said he was interested, but that he would write to he would ask his colleague, Peter Moore, to get in touch with me because they were collaborating on a project on the ribosome. And so that led to a postdoc at Yale. So again, I persuaded my wife and family to move yet again and crossed all across the US to the other side and spent a few years as a postdoc at Yale with Peter Moore, who was Jim Watson's graduate student, where he first encountered ribosomes and has been a world leader in the ribosome field ever since. And there, we were trying to look at where all of the ribosomal proteins were located in the small subunit. Now, the problem is just knowing roughly where each protein is on each of the subunits doesn't really tell you how a ribosome would work. I mean, if you want to understand how a ribosome works, this is not the way to do it. It's because the whole ribosome was very complex, and at the time, it was felt any information was better than nothing. So what is the problem? Well, the ribosome is an enormous object. It has 500,000 atoms, and it does this enormously complicated process of reading the genetic code, moving along the gene, bringing in tRNAs, stitching together the amino acids to make a protein, and knowing when to start and stop. So if you want to understand it, you really have to know what it looks like. Imagine if you didn't know what a car looked like. You were a Martian hovering above the Earth, and you just saw these little dots moving around in straight lines, or not so straight lines if you're in Chennai. So, the fact is, you wouldn't know how it worked, okay? So on the left is what a car looks like on the surface. But that gives you no idea how it works. Now, supposing you open up a car, then you start seeing how cars put together. You see that there's an engine in the back, in the back if it's a Ferrari, and you see a steering wheel, you see some stuff connecting, various parts of the car. You have a better idea. But then if you really look at things in detail, then you see how an engine works, how it transmits its power through the crankshaft to the wheels, how it's all controlled, how the steering works. Then you really begin to understand a car. And it's the same with molecules. Now, if you want to understand what a molecule looks like, molecules are very small. So how would you visualize something that's very small? How would you look at details? You would put a lens in front of it and magnify it, and then you'd be able to see it. That's what a lens does. It takes scattered rays from the object and recombines them to produce a magnified image that you can see, that you can look at details in. That's how microscopes or a magnifying glass works. But the problem is molecules are too small to be seen with a light microscope. And moreover, you cannot see the level of detail you need with a light microscope because the distance between atoms is much smaller than the wavelength of light. It's hundreds of times smaller than the wavelength of light. And in order to see detail, you need to use a wavelength that's roughly of the same order of magnitude as the detail you're looking for. That's a very old law in physics from the 19th century. So the way around this is to use a different kind of radiation, which is called X-rays. But the problem is there's no lens for X-rays. So how do you do this? Well, what you do is you take your molecule and you coax it to form regular three-dimensional objects called crystals, three-dimensional arrays called crystals. And then you take that crystal and hit it with an X-ray beam, and you measure the scattered radiation from the crystal. Now, because it's a crystal, the X-rays will only scatter along certain directions to produce spots. So the scattering pattern would look like that if you look at a detector. And in the book, I've tried to explain it. I should tell you, I've not tried to water down the science too much, but I've tried to make it so that the general reader, if they find it hard going, can skip over it or skim through it and still get a sense of what the science is about. So I hope you won't be discouraged by the science from reading the book. So, of course, I didn't know anything about this technique, which is called crystallography, but it was the way to get atomic structures of molecules. So I decided to go on sabbatical, and I wanted to do it in England because by that time I'd been in America for almost 20 years, and I thought it would be good to sort of get away from it for a while. And my wife and I, frankly, were Anglophiles in many ways. We liked Monty Python and English literature and all sorts of things, the Beatles. So we decided that Cambridge would be a great place because it was also the birthplace of molecular biology and of crystallography. So I wrote to Aaron Klug, who was a very famous Nobel laureate and also went on to become president of the Royal Society. And I was a complete nobody. And it shows you something of the character of the MRC lab and of Aaron Klug, that instead of, and you know, we've heard it, we've also seen it with Ramanujan, who wrote to G.H. Hardy, that instead of tossing my letter in the bin, he actually thought, well, that was a good idea, and he helped to sponsor me for a Guggenheim Fellowship and allowed me, which enabled me to go to Cambridge. When I came back, I collaborated with a colleague of mine, Stephen White, who was also from England originally, and we started looking at pieces of the ribosome and solved the atomic structure of individual proteins. But this would be like knowing what a spark plug looks like in detail, or a hose pipe, or the hub of a wheel, and without any idea of how they all fit into a car. And so it was really, after a while, quite unsatisfying. The alternative was to look at the entire ribosome. Now here, there was a history of the field, and that is that Ada Yonat, an Israeli scientist who went to Heinz Gunther Wittmann's institute in Berlin, she and Wittmann collaborated to produce crystals of the entire large subunit of the ribosome. This was an amazing feat because this is an object with, several 100,000 atoms, and larger than anything that had been crystallized before, except for viruses, which are very regular objects. And shortly after that, a few years later, a group in Russia, also headed by a woman, Marina Garber, crystallized both the small subunit and the large subunit of the ribosome. So now we had crystals of the large subunit, the small subunit, as well as the entire ribosome. Now, these crystals weren't of sufficiently good quality, which meant that you couldn't really get an atomic structure from them because the molecules in the crystals were not well ordered enough. But a few years later, Ada Yonat obtained crystals of the large subunit that did have sufficiently good quality. And I explain in the book what this means, sufficiently good quality. So in the mid-1990s, I was getting quite frustrated about progress on the ribosome. And the situation was that 15 years after the first crystals, there was still no apparent progress towards an actual structure. And there were good crystals of the large subunit, which is called the 50S, but no structure yet. And people were wondering whether this was really beyond the limits of the technique and whether it was too big to be solved. There were no good crystals of the small subunit or the entire ribosome. And I thought by this time, in the mid-1990s, the Yale group had decided that, Ada Yonat was not getting anywhere, and they decided to take the 50S crystals that she had obtained, which diffracted well, which had good quality, and started competing with her. And I thought, let the two of them fight it out for the 50S. I didn't want to get into that sort of competition. And I thought I would work on the small subunit, which didn't have good enough diffraction, but I had some ideas on how to make it diffract better. And I thought the whole ribosome was really too complicated and probably could wait until later. Now, so with that idea, I assembled a group at the University of Utah where I was a professor of biochemistry. I've only been a professor for about 3.5 years in my life. And so I persuaded 2 graduate students who are shown on the right. The guy with dreadlocks is Bill Clemens, who's now a professor at Caltech, which, by the way, wouldn't admit me as a graduate student in biology when I applied to them. And they've taken 2 of my students onto their faculty now. So, and the guy next to him is John McCutcheon. Now, these are 2 graduate students who knew nothing about ribosomes or crystallography, and that's why they thought that crystallizing the small subunit was a good PhD project, because they didn't know enough to know how difficult it was. So sometimes knowing too much is really a dangerous thing. And so with their help, we started this project. Now, soon after we got going, I went to a meeting in Sweden, and I got my first shock, and that was that the Yale group was making progress on the 50S, and Ada Jonat had, in the meantime, found a condition to crystallize the small subunit better. And so she had shifted her attention from the 50S to the small subunit, which is the 30S. So instead of competing with Yale and me being left alone in peace, I would now be in direct competition with her, okay? And she had a lot of experience and a huge group. She had 2 labs, one in the Max Planck in Berlin and Hamburg, and then another lab at the Weizmann Institute in Yale. And I had my 2 first-year graduate students and a technician, working on this, okay? So it was a bit of a shock, but I thought, well, she'd had crystals of the large subunit for, and had crystals for 15 years and really not done anything with them. So I didn't see why I should give up now because she had crystals of the small subunit. I thought the field needed new ideas. And then soon after that, Harry Noller, who was a ribosome biochemist and really with no experience in crystallography at all, hired the Russians who had crystallized the small subunit and the whole ribosome, and also hired Jamie Kate, who, these days he's also known as Jennifer Doudna's husband, because Jennifer Doudna has become very famous for CRISPR-Cas. But Jamie Kate is a brilliant and very well-known structural biologist of ribosomes. Anyway, he was one of the most brilliant people in the field, and Harry hired him as a postdoc. So suddenly, what was my quiet niche became a 4-way race, and it was a very stressful period that I describe in the book. So I decided in the middle of this race to move again, and this seemed crazy to most people. If you're in the middle of a race, why would you move? It'll cost you time. You'd lose at least a year in moving, and that's the worst thing you could do if you're in the middle of a race. I decided to move for long-term reasons. First of all, I wanted to work on whole ribosome structure, but I didn't know how long it would take. And in universities, you're usually funded by research grants, which are about 3 to 5 years in duration. Now, crystals of the ribosome had been around for 15 years, and there was no reason to think that it would be cracked anytime soon. And I thought, what if I'm working on this and I will, lose out? So anyway, I also felt I needed expert crystallographic colleagues, and the MRC lab, or the LMB, which plays a theme throughout my book, has a very collegial atmosphere. It also has an emphasis on long-term problems. It has a lack of hierarchy. It gives young scientists a great deal of independence and very early on. These are some of the reasons it succeeds. And it's really a general recipe for science. Science is really done when things are collegial, collaborative, when you give young people independence and freedom and so on. And I missed saying that yesterday about Indian science, and that's really one thing that's quite important. And it's an institute where there's a tradition of tackling hard problems, and the emphasis is on the importance of problems, not on how many papers you publish. So for example, Fred Sanger, who got 2 Nobel Prizes, only had 40 papers or so, and had what's called an H index of 18. Today, if you just went by his metrics, he wouldn't get tenure at most universities, and so it just goes to show you what is really important in science. So I picked up again, and unfortunately my wife agreed to my taking a 40%, or effectively more than a half, pay cut, and to go off to England right in the middle of the race. Fortunately, it all worked out. I don't have time to go into all the details. We ended up solving the 30S structure, while the group of Tom Stites, along with my former mentor, Peter Moore, solved the large subunit. And then we ended up solving the entire ribosome a few years later. And this also led to understanding how antibiotics bound to the ribosome, because once you had the crystal structure of the ribosome, you could solve that structure with antibiotics bound. And so we figured out how many different antibiotics bind to the ribosome. And that's actually leading to the design of new and improved antibiotics, including there are papers which show how these structures have been used to design better antibiotics that are effective against TB. Whether that will result in an effective medicine is not yet clear. That will have to await clinical trials. Now, the last third of my book is about the human side of science. Scientists are neither completely rational, like Spock in Star Trek, nor are they saints, okay? We're not completely altruistic. We're human beings. We're motivated by curiosity, but we're also motivated by ambition, by the desire to succeed, to be first. We have egos, we have insecurity, we have anxieties. I talk about all of these things and how people behaved when they were in the race, when there's also after the race, when there were high stakes. Rivalry and competition. Scientists are motivated, like all other human beings, by self-interest. We will collaborate or compete depending on what's appropriate. But humans are also altruistic. Many people also help me. They gave me lots of advice. They provided reagents, all without any credit, okay? And they provided technical help and so on. And none of them expected anything in return, like co-authorship or any of that. Although there were other people who really did do that. I also talk about the politics of recognition. This corruption starts early. We start giving prizes even in school, then we have bigger prizes. Ultimately, there are things like the Nobel. I talk a lot about what the Nobel is. Many people think the Nobel Prize means you're some sort of genius. It's not for being a great scientist. It's really for an important discovery or advance. And there are lots of factors. You could be lucky, you could be in the right place at the right time, you could simply be patient and persistent. It certainly doesn't mean that you're a genius, and it certainly doesn't mean you know anything outside your own field. And also how people behave when stakes are high. And this, of course, led to what I call the ribosome roadshow. So after the structures, lots of conferences all over the world, and you could see all these people, almost like politicians campaigning, talking up their work and slightly talking down other people's work, and jockeying for the 3 chairs when the music stops. I'll be right down. And, the auditions multiple times in Sweden and picking an argument with a guy who ended up on a committee. So I was pretty sure I would never get the Nobel Prize. And so when I eventually got the phone call, I thought it was a prank. And I'm still amazed, given my background and history, that this is sort of where I ended up at this, Nobel banquet in Sweden. Now, there are Nobel Prizes in physics, chemistry, as well as in physiology and medicine. But the newspapers realized that of all those prizes, actually the important thing was the ribosome, because the next day on the front page, this is what they featured, Tom Stites and me. Clearly, they understood the importance of the ribosome. And the fact that Princess Victoria was in the middle had absolutely nothing to do with the photographs that they chose. So there's some general lessons. Keep your options open, even if it means learning completely new techniques, not being afraid to ask for help or show your ignorance. You have to talk to people. And also, everybody has imposter syndrome. I always felt I was constantly fooling people that I didn't really belong there. And somehow I would eventually be found out and they'd realize I really wasn't that bright and didn't know anything. And so this is a natural thing that people have. And I think people who are outsiders feel it more. And for example, women who are a minority in science will feel that more. So I just think that people who feel that way should realize this is a very common thing and you shouldn't worry about it. And of course, success depends on many things, including luck and patience and skill, and of course, money for the science. So I was going to show you the movie, but I think I would rather take questions from the audience instead of showing a movie. But if you Google ribosome translation movie on YouTube, you can see a movie about how ribosome works. And in the time that I've been talking to you, the thousands of ribosomes in the trillions of cells in your body have all been churning out thousands of proteins from the genes that they're reading. So thank you very much, and I'll take questions.
It's a very inspiring, very fine talk.
Now, you have to realize I'm deaf. I have hearing aids.
Okay.
So you need to talk loudly and slowly.
Okay. It's a very fine, very inspiring talk. And like Rutherford, you have got transformed from physicist to chemist, physicist to biologist, and you won Nobel Prize in chemistry, which looks very strange for us. You started as a physicist, right?
Yeah, I think these borders are somewhat artificial. First of all, chemistry is what is called a bridging discipline. So biology is, the functioning of biology is done by molecules. So if you have a biological molecule, often the Nobel Prize will be in the chemistry category. So I think these distinctions are somewhat artificial because the boundaries between these disciplines are not very hard. They're really blurry. And so you shouldn't make too much of that. I know in India people think that's a very big deal, but actually it isn't. You just need to follow the problem.
Also, one more question.
I think,
Good afternoon,
in the interest of fairness, we need to limit each person to one question.
Good afternoon, sir. So we are living in the times of gene editing.
Sorry?
Good afternoon, sir. So we are living in the times of gene editing and designer babies. So there are varied viewpoints around this, such that on one hand, it's said to boost the immunity of the baby by making it immune to deadly diseases. On the other hand, it's considered blasphemy as it defies nature. So what are your views on the ethical concerns of such practices in the present and in the future?
Of gene editing. Yeah, so I think gene editing is a complex problem. First of all, I'll answer this one question, but I'd rather other questions refer to my talk. This has absolutely nothing to do with my talk, okay? But it's an important problem, so I'll address it. Gene editing is a complex technique which allows us to correct DNA information, any mutations in our DNA. It has to be used ethically. There have to be proper guidelines. There have to be studies on safety. But in principle, I don't see why it could be stopped indefinitely. Because if you have a genetic disease, let's say you're a parent, you want to make sure your child is free of a genetic disease, then you would want to actually be able to correct that genetic defect. So I think this is a problem that society needs to solve, not just scientists, but scientists with the consent of society at large, with the informed consent, with proper regulation.
So, isn't RNA found only in prokaryotic cells and DNA found only in eukaryotic cells?
No. Both eukaryotes and she's talking about the difference between bacteria and single-celled organisms like bacteria, and organisms which have a nucleus, which are like us. So the difference is, there is no difference in that sense. We both have DNA and RNA, and DNA is copied into RNA, which is then read to make protein in all life forms.
Thank you, Dr. Venki, for that fascinating conversation that you had with us. My question to you is, your journey as a scientist has been almost like a ribosome. You've codified the science of gene, and then you have translated your understanding of crystallography, and then stitched up your own work of science, which has won you the Nobel Prize. Now, what I realize in your life journey is that you've taken multidisciplinaries and been able to concoct your own understanding of gene science, and then built up your own platform. Is it viable that as scientists and as professionals, multidisciplinary subjects, taking them gives you a broader perspective of understanding implications of a certain problem?
I think this is complicated. I think in my case, I didn't think of it as multidisciplinary. I simply followed, my next goal from one thing to another. I mean, I left physics because I didn't think I was going to be good at it, or I felt I didn't have the right background. And so I didn't deliberately set out to be multidisciplinary. What you do in science is you pursue a problem and you use techniques whenever you can. Sometimes they cross traditional disciplines and sometimes they don't. There are people who work within a discipline and have done brilliant, great work. I'm not sure that it actually gives you a broader perspective.
Hello, sir, congrats once again on your Nobel, and I actually request your reflections on the critical impact of genetic engineering on human and animal life. Particularly, I mean, like we have cancer trains running in Punjab, and insulin resistance issues, and then air pollution in Delhi. Like they're coming back to animals, particularly in cow. We are making the cow to yield 100 liters than 20 liters, and also reducing the life from 20 years to 5 years. Now assume somebody does the same thing with human beings.
I'm sorry, I didn't really understand.
I'm just asking
you
about
have
the
to talk
impact.
a bit slower.
Oh, sorry.
Yeah.
I'm trying.
What is your question?
This is impact of genetic engineering on human and animal lives.
Changing what?
No, critical impact of genetic engineering on human and animal lives.
Yes.
Like we can see in a lot of things happening in everywhere, like in Punjab, we have cancer train running between Punjab and this.
Are you suggesting that the increase in cancer is due to genetic engineering?
Yeah, exactly, correct.
Yeah,
I
completely disagree, okay?
No, you can agree, but all the doctors...
I'm sorry, this is a myth that is, there are, I should say, groups that are against multinational corporations, and they're confusing their hostility towards multinational corporations, monopolistic practices, etc., with the technology. This would be like saying you're against knives in the kitchen because there are people who commit murder with knives, okay? So I think, I'm sorry, first of all, this has nothing to do with my book, okay? I'd rather questions be limited to my talk, okay?
Good afternoon.
One last question. They said,
Yeah,
sir.
The question is,
sorry.
Just a second. So the question is, the reading of this ribosome here. So when the ribosome is reading from genetic code, is there a possibility that it can do a wrong reading? So is it always right?
Very good question. The ribosome often makes mistakes, and some of the antibiotics that work by making the bacterial ribosome make more mistakes so that the bacteria effectively die. So, it is absolutely true. And there's always a compromise. Just as with typing, there's a compromise between speed and accuracy. And the ribosome has evolved to have the optimal compromise between speed and accuracy. If you make it more accurate, it would be too slow. And if it's too mistake prone, then it would be deleterious to the cell. So somewhere in between is the optimum. Okay. So I'm sorry, I have to.
Can I ask
I've been told
Excuse
to
me,
finish up.
one question, sir.
Can I ask one? Last.
Sir, only one question.
So can I ask?
Excuse me.
I've been
Sorry,
waiting
one
for some
question.
This guy has been standing up
Okay,
for
after
many
that, I will come.
minutes.
Actually, I read Francis Collins' book on this, who was part of the Human Genome Project, The Language of God. So what
Sorry?
is the language of God? He calls DNA the language of God. Francis Collins,
Yes.
in his book, who was part of the Human Genome Project.
Yes.
So where does God come into this?
Well, you'd have to ask Francis Collins. He's a very religious man. He's a very religious man, and I'm unfortunately not. I would say it's the language of evolution. Okay. Thank you. I'm permitted to take one last question. So if somebody has a burning question.
Excuse me, sir, only one. Sir, my profound respect to you, Professor. My profound respect to you, Professor. Only one question.
Yes.
Craig Venter, after human genome, he commented, "You are not what your genes are."
Yes.
Can you please comment on it?
Yes, so I think, people think of genes as a blueprint, but that is a mistake, because if you take a blueprint and build a machine according to the blueprint, it comes out identical every time. So if you have a blueprint for a car or a house and you use the blueprint, it'll look exactly as the blueprint defines. You have to think of DNA more like a recipe, or a music score, okay? So if you have a recipe, and if 5 people use the same recipe, it's not going to come out exactly the same. The cake won't look exactly the same. And you can ruin the cake. You can either ruin the cake, or if you're a very good cook, you can actually make an absolutely delicious cake, and you might actually enhance on it. So so genes convey a predisposition and a potential, but they're not by themselves completely destiny, okay? So it's a complicated question. But that is not to say you should dismiss genes. Everything is not nurture. People know, identical twins are more similar than they are, to other people. We're more similar to our relatives than we are to some random person. So genes do play a very important part, and we know that from selection of plants, from breeding of animals, and so on. That genes are very important, but they're not destiny. It's a complicated interplay between genes and nurture that makes us who we are.
Thank you, Doctor. Ladies and gentlemen, kindly do not crowd the author as he leaves the stage. He will be available for book signings and interactions outside the auditorium.